Hey everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGiorgis. The paper we're going to talk about today is titled Mutation in the Alpha-Synuclein Gene Identified in Families with Parkinson's Disease. This, of course, is part of our online PC Neurobiology class, 2020. The paper begins by talking a little bit about Parkinson's. They say, Parkinson's disease was first described by James Parkinson in 1817. The clinical manifestations of this neurodegenerative disorder include resting tremor, muscular rigidity, bradykinesia, and posterior instability, and of course, ultimately death. It turns out at autopsy, a key feature are these lesions that are called Lewy bodies, and they're found in many areas throughout the brain. Finally, while most cases of Parkinson's disease are considered sporadic, that is, they occur from unknown reason, about 5% are heritable. That is, there's a genetic mutation in a gene that causes the disorder, and that's what they're looking at in this particular paper. Now, they also have a pedigree and an inheritance pattern for this disorder in a large Italian family, and I'm just going to draw the pedigree in this case because there's a little bit of a wrinkle. So men or males are indicated by a box and females by a circle. And this couple had a daughter, two daughters, and a son. And afflicted individuals are denoted by filled in circles or squares. So this daughter is afflicted, and this daughter is afflicted. It's interesting to note that the parents, neither of the parents are afflicted. And this daughter gave rise to two sons. And both of the sons are afflicted. Then this son has a son who's afflicted, and this son has a son who's afflicted. This son has a son who's not afflicted and a daughter who's not afflicted, who marries a person who's afflicted, and their son is afflicted. This son had a son who's afflicted, and he had a son that was afflicted and a daughter who was afflicted. And this son married an unafflicted woman, and they had an afflicted daughter. This woman married a man who was unafflicted, who was the son of an unafflicted man, who was one of the sons of this woman, who had a second son who was unafflicted, and a daughter who was afflicted. So this is a little bit of, I don't know, you'd have to figure it out, but obviously these two people married and they're pretty closely related. Anyway, this daughter was afflicted, and this son had a daughter who was unafflicted, and this woman had a son who was afflicted, who married a woman who was unafflicted, and they had two sons that were afflicted. And these two people had two sons that were afflicted. So I drew this out, and I'm sorry it took so long, because I said that this is autosomal dominant, and you'd say, okay, why is it autosomal dominant? Well, you'd say it's not recessive because it never skips a generation. But this woman here has a son that's afflicted, but she doesn't have the disorder. But obviously, this son received the gene from his afflicted father. So it still holds to the rule that the disease doesn't skip a generation, although the parents, neither of these, or the original couple, neither one of them had the disorder. But it doesn't skip a generation, so it's not recessive. It's not Y-linked because both men and women, females and males, have it. It's not X-linked dominant from the mother because all sons that are afflicted must have a mother who's afflicted, which isn't the case here. And all daughters of a male that's afflicted must be afflicted. And there's this case as an example. Is there another case? No, I guess that's the only one on the board that I can see. But at any rate, it's an autosomal dominant inheritance pattern. So just to summarize again, Parkinson's disease is characterized as a neurodegenerative disorder that includes resting tremor, muscular rigidity, bradykinesia, and posterior instability, and of course, obviously, often leads to death. It turns out that there are some forms that are heritable, that is, there's a mutation in a gene that causes that disorder, and in this particular case, the mutation that they're looking at follows an autosomal dominant inheritance pattern, and at autopsy, individuals that have Parkinson's disease have brain lesions called Lewy bodies. This heritable form of the disorder is also an early onset form with the age of onset at 46 years plus or minus 13 years. They also tried to calculate the penetrance of the disease by studying the pedigree, and they estimate the penetrance to be 85%. Now it turns out the way they found the mutation and the gene responsible for Parkinson's in this family is by using positional cloning. And we've gone over this before in the Cartier-Harlan paper, and that's how they found the mutation in the amyloid precursor protein that caused Alzheimer's disease in the family that they were studying. So I'm not going to go over that technique again. If you want to review that, please look at the video, Cartier-Harlan, and check it out. But look, positional cloning is a type of linkage analysis. Even though we're extremely similar genetically, we all have differences with the obvious exception of twins that are genetically identical. But we all have differences, and if you know of those differences and where those differences lie on the chromosomes, then you can follow those differences that they refer to as markers in the paper. You can follow whether that particular genetic difference co-segregates with the disease. And if a marker of known location segregates with the disease, you can then determine roughly the location of the mutation that's causing the disease in that family. So instead of using the terms marker and genetic differences, in the example of Cartier-Harlan, I was trying to use trait because, of course, different traits are made by differences in the genome. So, for instance, if you knew that hair color was caused by a single gene on chromosome 2, and every time a person had brown hair, they also had the disease, that would suggest that the gene for brown hair and the gene that causes the genetic mutation are very close to one another on the same chromosome. And through that linkage analysis, they were able to figure out that the gene responsible for Parkinson's in this large Italian family was located on chromosome 4. They say in the paper that they also knew that this protein called alpha-synuclein, which is also involved in Alzheimer's disease, is located in that region of chromosome 4, and for that reason they suspected that a mutation in alpha-synuclein may be causing this form of early onset Parkinson's disease. So once you have the nearest marker that co-segregates with the disease, you simply begin sequencing in both directions along that chromosome from where the marker lies, both in the 5' and 3' direction of that chromosome. And through that process, they ended up finding a mutation in exon 4 of the gene. It's a single point mutation that changes the nucleotide G to an A at position 209, and it causes an alanine to be changed to a threonine. Now, once you find a mutation like this, you need to prove that the mutation actually causes the disease. So you need to determine whether all of the individuals that are afflicted with the disease have the mutation, and individuals that don't have the disease don't have the mutation. It turns out that this particular mutation added a restriction site to the sequence of that afflicted allele. So this is the opposite scenario to what we looked at in yesterday's paper on fatal familial insomnia. So let me go over the genetics of that and what they're showing in figure 2. Okay, so if you have a double-stranded DNA molecule, a chromosome, of course this goes on for a very long time in this direction, and it goes on for a very long time in this direction, and they've sequenced this region of chromosome 4, so they know the sequence, they have that, and they know where the mutation is. Let's say it's right here, and all they have to do is make PCR primers that flank either side of the mutation, one that will bind here and one that will bind here, and in doing so you will amplify this region of the chromosome. And of course, there are 2 copies of chromosome 4, and one of those copies is going to have the mutation, and one copy does not have the mutation. But these same 2 primers that you've designed are going to bind to both chromosomes, and they're each going to amplify this same stretch of chromosome 4, except that one has the mutation here and one doesn't have the mutation. So if you have a gel, which they show in figure 2, if you PCR amplify, you have a test tube, you put in some cells that contain the genome, in the nucleus, and you put in the PCR primers and the reagents required for the PCR reaction, and you then run the reaction, you are going to amplify, again, 2 bands. You're going to amplify, oops, I don't know why it jumped like that. You're going to amplify this band and you're going to amplify this band. And they say in the paper that the distance between this primer and this primer is 216 base pairs. And if you run a gel, then you have to treat this with the restriction enzyme that turns out to be able to cut this form of the gene, because with the mutation it created a restriction site, and the enzyme can now cleave the mutated version of this region of alpha-synuclein. But it won't cut this region here because it doesn't contain the correct sequence for enzyme recognition, for recognition by the restriction enzyme. So if a person harbors this mutation and you use their DNA in a PCR reaction and you cleave it with this particular restriction enzyme, which by the way is called TSP45, that's the name of the restriction enzyme, you should get one copy of the wild type version, and that version is 216 base pairs long. And then you should have one that gets cleaved, that is, the mutated allele should get cleaved, or the mutated PCR product now should be mutated, and you should get a band at 128 and 88 base pairs. So you get another band here at 128, and then a band down here at 88 base pairs. Okay? Now, if you don't have the mutation, then you're just going to have two copies of this version that is uncleaved. So just kind of a heavier, slightly heavier band, because there's two copies of the 216 base pair band. And so now you can just take all of the individuals in the family that you want to test and put some cells in the test tube for each of those individuals. 1, 2, 3, and so on. After the PCR reaction, add the restriction enzyme and incubate it for an hour or so to allow the cleavage to happen. And then run the gel. Run the PCR reaction out in a gel. And if you get 3 bands, then you know that the person has one good copy and one bad copy. And if you just get one band at 216, you know the person should be wild type. So, if indeed this mutation causes the disease, then every person that has the disease should have this restriction pattern. And anyone who's wild type should have this one. This is wild type and mutant. Okay? So I'm going to go back to the pedigree so that we can try to analyze figure 2, which is the gel that they ran on the individuals of the pedigree. So now if you look at figure 2, looking at the gel, first of all they analyze the DNA from this person. This person, which is number 1, this person, which they say is number 3, this person, who is number 30, this person, actually, this female, which is number 4, this person who is 19, this person who they say is 6, 5, which must be this person who's wild type, or unafflicted rather. Then they do this person who's 23, this person who is 24, this person, this person, who is number 7, this person who's number 8, and this person who's number 9. Okay, sorry, it's a little sloppy. And they, of course, do PCR and then the restriction enzyme. And in lane number 1, you see that this individual has the band at 216. That person has a band at 218. And they have a band at 88. So this person is afflicted. They have both alleles, one mutated allele, one normal allele. 3 is an individual who's unafflicted, and they have 2 copies of the unmutated version of the protein. Now, 30 is a little weird because, as we said before, 30 is the son of this unafflicted person, and that can't happen in an autosomal dominant disorder. If this male is afflicted, their mother must be afflicted. if they got the disease gene from her. But in this particular case, a male came along and they had a son that was afflicted, and that mutation comes from the father. So it turns out number 30 shows up as the two wild type versions of the protein. 4 is this person here. They have 2 copies of the wild type version. 19 is this afflicted female. So it turns out that she shows the mutation. 6 is this mutated, 6 is from this afflicted male. 5 is from an unafflicted male here. 23, 24. Anyway, the pattern is the same throughout, that all of the individuals that are afflicted with the disease, with the exception of 30, has the triplet, has the mutation. And those that are unafflicted do not have the mutated form. They only have this wild type version of the protein. So they go on to look at other families that have a heritable form of Parkinson's disease, and in this case they find a number of Greek families, and they show in the second part of figure 2, they analyze those families both in terms of the pedigree and in terms of the phenotype, and in those individuals, those people that are afflicted have the mutation. It also turns out that they tested individuals from a tissue database that are wild type. They're just people that live in Italy from a blood bank, and they were able to show that all of those wild type individuals lack the mutation. So pretty strong evidence that this mutation in alpha-synuclein causes this heritable form of Parkinson's disease. I should also mention that whatever mutation this is that causes the disease in this individual and this individual obviously must be a different mutation than the one that all of the other individuals in the family that are afflicted possess, because we didn't get 3 here even though this person is afflicted. So a different mutation either in the same gene or in a different gene. They don't go into detail over that particular issue. Okay, there's one last experiment. They wanted to figure out whether the diseased allele was actually expressed. Now, we've said this many times before, that the disease gene must be expressed in an autosomal dominant pattern, if you have an autosomal dominant inheritance pattern, because the diseased allele is the thing that's making you sick. So they just want to prove this to be the case, and I'll draw this diagram out for you as well. Okay, so in the paper they say that they purify RNA and they reverse transcribe it from a lymphoblast cell line that was created from one of the members in the Italian family. So again, you have to take total RNA, purify the total RNA in this case with the poly A tail. You have to bind a set of T's to the tail and use reverse transcriptase to make a DNA complementary strand to the RNA molecule. Then you dissolve the RNA molecule with an RNase, and you add DNA polymerase, which makes a complement to the complement, and now you have a double-stranded double-stranded DNA molecule in which the top strand is identical to the RNA molecule, except the U's have now been replaced by T's. And then they use that. They design PCR primers, again, one that binds here on one side of the mutation, on the 5' side of the mutation, and one down here on the 3' side of the mutation, and you amplify this region here, and then they once again cleaved it with the restriction enzyme. But in this case, they're looking at RNA sequence rather than genomic DNA sequence, which they used to analyze the pedigree in the large Italian family. Okay, so in figure 3 they show you the gel of the PCR reaction for amplifying RNAs from both wild-type individuals and individuals that are afflicted and presumably harbor the mutation. The question is whether that mutated allele is actually expressed as an RNA molecule. So they design these PCR primers, as we talked about, and that will amplify the RNA or the cDNA in this region from this PCR primer to this PCR primer. And you could also obviously have a version that's wild type. So you could have one that's afflicted, or one that's mutated rather, and one that's wild type. And they say that the PCR primers are designed in such a way that the distance between the two primers and including the primers is 500 base pairs long. So 500 base pairs. So in their gel, they show, first of all, the first lane is just a molecular weight marker, and that's used to estimate the size of your PCR products, so that it just has some bands of known molecular weight on it like this, and some of the bands are pretty faint so you can't see them. But at any rate, if you take a wild type person, which happens to be in lane 2, then they get a band that's 500 base pairs long. So this is 500 base pairs, okay? Which makes sense because you're just amplifying a wild type individual from here to here. That's 500 base pairs long. And this is not digested. So all they're doing is doing the PCR amplification and showing you that the product is 500 base pairs long. That's it. Okay, that's lane 2. This is wild type. Oops. This is wild type. Okay. Then in lane 3, they have wild type, but they add the restriction enzyme to digest the DNA. And it turns out that there are some restriction sites within that 500 base pair band because the DNA does actually get cleaved. And when they cleave it, they say that they get a band that's 249 base pairs long and another one that is 218 base pairs long. The problem is I added these two up, and 249 and 218 is a 4. That's 7. I said 4, 5, 6. That's 467, and that doesn't equal 500. So I stared at this thing for a while because I just couldn't figure out what the problem was, and it must be that there's another restriction site that cleaves this 500 base fragment that must be 33 base pairs long. But what they get is they get a band here at 249. They get a band here at 218. And then if you look down here, you can't see the 33 base pair band. And there's two explanations for that. One is that it runs off the bottom of the gel, so it just ran out the bottom into the solution. Or it's so small that you just can't detect it. There's not enough DNA there in order to see the band. But at any rate, that must be the case because the math isn't working out. Now if you look at an afflicted individual, the RNA of an afflicted individual, in individual 4, they again just do a PCR reaction and they therefore get a 500 base pair band. That is, there's no restriction enzyme. They're just showing that as a control, if you do the PCR reaction, you get a band, and it's of course the same size as this band because both the mutant form and the wild type form are the exact same size. They're both 500 base pairs long, and the question is whether you end up detecting the mutation. So then they do a experiment where they do the PCR reaction and then they add the enzyme. What was it? TSP45, I think. And that cleaves both one wild type version, because the idea is that an afflicted individual has one wild type and one mutated form of the RNA molecule, and they get a band at 249, at 218, and then you get a third band which is at 185. So that's weird because the math, once again, doesn't really add up. So we have a band that's at 249. We have a band at 218. And now we've figured out that there must be a band that's 33 base pairs long. And in this lane here, you have a band at 218. You have a band at 33, which runs off the bottom of the gel. And we have one band at 249. And the wild type should cleave this way. The question is, in the mutated form of the RNA molecule, we would have a 33, we would have a 218, and then they have a version that's 185, which gives us another piece that's 64 base pairs long, because the band that's cleaved must be one of the two bands. I mean, there's two copies of this 249 here, and there's two copies amplified in the reaction, but in this case, one of the two copies must be cleaved to generate the 185 base pair band, and that gives us a remainder of 64 base pairs, which again must be run off the bottom of the gel. That's what I think. They don't mention it in the paper, but I think that's what's going on. At any rate, this shows that when you amplify the RNA from an individual that's afflicted with Parkinson's disease, you get two different RNA molecules. You get one that's wild type, and you get one band of 249, one band of 218, and one band of 33. And then in the mutated form, you get a 218, a 185, and you must have a 64 and a 33 that's run off the bottom of the gel. So it shows that the RNA molecule is obviously expressed, and the expression of this mutated form causes an early onset form of Parkinson's disease, a heritable form. Okay, you guys, that's it for today. I hope you guys are doing well, and I'll talk to you tomorrow. Bye-bye.